Thank you, Erica. Thank you for the introduction and also inviting me to give this talk. I will be taking you back to the first year of my PhD in 2021 when I started working on mushrooms with mass spectrometry imaging. Sorry, my slide's not moving. Oh, yeah. So before I talk about mushrooms, we are a part of AMI node within Euro-BioImaging, and we have different groups within Maastricht University having different facilities like optical microscopy, microvision imaging, advanced light and electron microscopy. And we are open, and you can always contact us to use these beautiful imaging techniques. And we are in the center of Europe, here in Maastricht, which is in the south of the Netherlands. And now coming to the core lab of mass spectrometry imaging here in M4I. So CORE stands for Collaborative Open Research and Educational. We collaborate with everyone to push science forward. We are open and accessible by everyone in the world. Michiel van Bosch, he's the core facility manager and also has his own small group. He's also a PI, and you can contact him if you have any samples that you want to analyze for metabolites, lipids, proteins, peptides, sugars, polymers, and many more. And not only for these molecules as quantification or identification, but also to look at their spatial distribution within your samples. And then we are a part of Maastricht University, so we do both research and education. And we have Ruben Jacobs, who is a research technician in Core Lab. And we have a lot of instruments related to microscopy and mass spectrometry that you can use, or send your samples and we will analyze them for you. So coming back to food and imaging, we all are very familiar with such labels. We look at carbs or lipids or proteins of our food. And how do we get the identities or quantities of those molecules? One of the very common technique, maybe some of you are using alongside your imaging techniques, is liquid chromatography mass spectrometry. And you get the composition of your food. However, as you see a bottle of juice here, you have to homogenize your tissues or your samples, so you don't get a spatial distribution. So what I did in my PhD was looking at the spatial distributions of molecules by mass spectrometry imaging. For an example, there's a slice of carrot, and it has different anatomical structures. And I can show you the 3 different colors here, which show 3 different molecules. For example, a sugar, a fat molecule, or a protein in the carrot slice. I will talk more about how we do that in a minute. Before that, I just want to give you a brief idea about what else I did besides mushrooms in my PhD. So mushroom is what I'm going to talk about in a bit. I also analyzed burgers for different ingredient classification that was combined with classification models. And then I also looked into plants and plant microbe relationships. And besides my thesis, as everyone else does, I've tried different other experiments. This is an image of sweet snap showing some of the lipids and small metabolites in different colors. This is a bacterial colony. And as I was a part of Marie Curie funds, I went to a research secondment to Barilla in Italy and did some mycotoxin analysis there and brought back some of the wheat kernels. And what you see in the corner here is, maybe I'll take out my laser. This is an image of wheat section. It's a 20 micron meter section, and the green, blue, and red are different lipids in the wheat section. So now mass spectrometry imaging, one of the common techniques in MSI is MALDI, which stands for matrix assisted laser desorption ionization. And I mainly use this in this publication or this chapter of my thesis. So how do we perform MALDI? We take our sample and we have to make very thin cryo sections within micron meter ranges. So usually the biomedical samples are even lower than the food samples, like 10 micron meter. But in my case, mushrooms had 90% water, so it was difficult to have that thin sections, and I could reach 17 micron meter. So we mount the section on a glass slide, and then we spray or sublimate a matrix on it. Matrix is a chemical compound which we dissolve in a solvent, and the solvent can be polar or non-polar, considering the target compounds. If they are more polar, then we take the polar solvents. If they are non-polar, then we take the non-polar solvents. So we mix a chemical in a solvent and we spray it on our slide, and we have a robotic sprayer that does the job. So then you see there's a layer of matrix on my tissue. And what matrix does is it co-crystallizes with the analytes of the sample and helps in ionization of those molecules or analytes of our interest. And it helps in absorbing the laser. And why I'm talking about laser is when the sample is introduced into the mass spectrometer, the laser shoots on the sample pixel by pixel, generating ions which go into the mass spectrometer. And from each pixel, you get a mass spectrum. And in the end, the mass spectrum from all the pixels are combined. Here, there's an example of three different compounds. So mass spectrum is mass to charge ratio versus intensity. So we are here measuring the mass of electrically charged molecules. So that is what is mass to charge. And so the peaks here in green, red, and blue are showing three different compounds localized in three different regions of the mushroom. And if I combine all these three RGB images, then I get the spatial distribution of all the three in one image. So what I found among the flavor profile, I will show you that in the next slide. Before that, a brief introduction about the taste compounds in mushroom. So we all enjoy mushrooms, and especially the umami flavors, but there are also other flavors like sweet, bitter, salt, and sour. And these are contributed by molecules such as amino acids, soluble sugars, organic acids, salts, and nucleotides. So what I did was I made cross sections of fresh frozen mushrooms. However, we don't always just eat the fresh mushrooms. It's good to know the distribution in a natural state, but we often cook them or bake them, or they're on our pizzas and soup. So it makes sense to also look at heat-treated mushroom. What happens here is some of the compounds, which are amino acids and are very important for taste, they are low abundant, and it is difficult for multi-MSI to ionize and detect them. So I added a chemical compound, which we call as on-tissue derivatization. We spray it with another compound, just like we sprayed matrix, and that adds a positive charge, in this case, a mass, which is positively charged, 177. So I used a compound called TAHS, and then the ionization is boosted, and I have an added mass on top of my molecule, so it's easier to detect. So some of the results, although I have many other taste molecules that I detected, but few of them I'm showing here today. So glutamine is one of the umami tasting compound, and you see in the fresh frozen mushroom, here, according to the intensity scale, the yellow or white color means higher intensity. So in the fresh frozen mushroom, you see higher intensity of glutamine in the gills, while you still see some here in the heat-treated mushrooms, but probably the property of these small molecules to dissolve in water and then go away with heat or get evaporated might be the cause of lower intensity after heat treatment. And then a very cool distribution of a bitter tasting tyrosine compound. and a sour tasting malic acid, and a sweet tasting glycine. As I said, there are many more amino acids and organic acids that I analyzed. However, lipids are also very important part of our taste profile because they are carriers of flavor and they are odor active compound. So I looked at lipids, and sometimes there was not a very big change, but in other lipid molecules, you can see an enhancement in the signal of the molecule. So it could be that when we are heating, we are enhancing the flavors, and that's why we might enjoy the mushrooms with which are heat treated or cooked. You might ask that if these methods are just for a white button mushroom, which was in the previous slides, I will say no, they are applicable for many other mushrooms. One of the examples of chanterelle mushrooms, which I worked together with a group in Technical University of Munich, and again brought back some of the mushrooms to my lab to image. And you can see that we are able to sorry, work with the same method, and I can see the compounds in the sections of chanterelle mushrooms. The mass spectrum shows the similarity of the two mass spectrum for chanterelle and white button mushrooms. And here are the images showing some of the similar compounds that you saw in the white button mushrooms. Now, we aim to help food industries in extracting these taste compounds from particular structures. And there are already companies, and some of them are in touch with us for not preferring chemically derived taste molecules, but to use such options such as mushrooms to extract taste compounds. And then we also know that the mushrooms can be harvested and grown in different conditions and harvested at different times to get different taste profiles in the different structures of mushrooms. So I think imaging will also help the growers to do that. So I have an example of another technique because MALDI also sometimes have issues with the sample preparation. And in this technique, you will see that I don't need any sample preparation. And it's very cool. Going from right to left, we have a laser, which is a carbon dioxide laser. And we connect it to very close to our sample, and the sample is directly connected to a sample holder. So I just took out the mushroom from a box and put it here. And then we connect a tubing from the laser towards the mass spectrometer. And this is a computer that's connected to the mass spectrometer, and it will show us the mass spectrum. And there's another computer that controls the laser. So how we do it, I can show you a quick video. Now the laser will shoot somewhere here, and you will see smoke getting through the tube. You won't see the smoke getting through the tube, but there's a cloud of smoke that comes here every time it shoots. And we have told the computer to make these patterns and shoot laser in specific patterns. And then one of our colleagues helped in making a software based on MATLAB that can give us the molecular profile in the 3D of mushrooms and many other samples that we tried. And here's an example of lipid. And you can see according to the intensity scale, yellow is lower intensity and blue is higher. So where in this circle, the lipid is lower or higher intense. So this 3D scanning gives a great map of the molecular gradients from surface to the core. We can make changes to where we want to shoot and make patterns. And sometimes in the processing industries, we need to have food that is more crispy on the outside, but have a certain other texture on the inside, or maybe a uniform texture that we are looking for. So such instruments can help if they're a part of their processing lines. We have So yeah, so they can smartly control the process and give us consistent product quality. There was no sample preparation, as you saw, and the measurements were very quick. Each hole we made was 2 millimeter and took 5 seconds to shoot and give us a mass spectrum of it. I would like to thank my supervisors who guided me, helped me, and supported me during my PhD, and my colleague Bryn, with whom I learned a lot. This is our group in M4I. And I thank Futranet, which was the Marie Curie Funds, Barilla, and Technical University of Munich. And thank you for listening.